So the first few things we're going to talk about, we're just going to cover some genetics jargon, just some of the vocab that you guys will need in order to be able to understand this material. We know the central dogma, so we're going to just fly right past that. You know the characteristics of DNA and chromosomes, especially bacterial ones. We've talked about how that is different than a eukaryotic chromosome. So we'll fly past that. And we'll look at replication, transcription, and translation. And if we have time, we'll start the second part of chapter 9, which is looking at when things go wrong. So when we're talking about mutations and how those can affect organisms. And then we'll talk a little bit about regulation of genes, which we briefly covered when we talked about regulation of enzymes. So just some genetics jargon. Also, I apologize, your notes are missing a few words, like 4 total in the entire PowerPoint. I'm sorry. So they're blue, just so that you can write those down.
Oh.
So genetics, pretty simply, is the study of genes. So looking at DNA, DNA is the message that is going to basically tell the cell what to do, how to look, how to replicate, when to replicate, what environment it likes. Everything that makes up the cell and who it is thanks to its genes. So we're going to be looking into that. Very similar to us, genes are going to determine characteristics. So all of us look different from one another because none of us are genetic clones of each other. We are all different genetically. And that's the same for a lot of bacteria. Genetically, they are different enough to give them different traits. The genome, when I say something about E. coli's genome, what I'm talking about is the sum of all of the material that is found in the cell that contributes to genetic information. So the chromosome is different from the genome. And that is because there are extra chromosomal units of DNA sometimes found within a cell. So yes, every organism is going to have some kind of chromosome, but we can have things like plasmids that are going to code for other information. So when I say genome, I'm talking about everything that is genetic within the cell. The chromosome is the structure that carries most of the genetic information. So in our case, the bacterial chromosome being that circular piece of DNA that is found inside of the cell. A gene is part of a chromosome. So a gene is a very small unit found within the chromosome that is going to code for something. And you guys should be pretty familiar with that information. The genotype is the genetic makeup of an organism. So anytime we characterize something based on its genetic makeup, we're talking about its genotype. And any of the traits that we are able to observe thanks to those genes are the phenotype. So for example, a genotype, I could say, I don't know where the gene for your eye color is, but let's say that I said, wherever your gene for your eye color is, the nucleotide sequence looks like this. You know, T, T, A, C, G, blah, 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 blah. Go on and on and on. I could say that. That would be the genotype, the genetic makeup for a potential property, eye color. The phenotype would be you have brown eyes, you have blue eyes. So that is what is actually expressed and observed thanks to the gene. Does that make sense? Okay. And genomics is this newer field where we can really start to now look into the genes of a particular organism and start to characterize it based on its genetic information. So up until very recently, we were not able to really sequence somebody's DNA, whether that be an organism or a human or whatever that might be. It was very recently that we actually said, okay, now we can look at your DNA and we can tell you exactly what that sequence looks like. And we can tell you something about yourself based on that sequence. The first time that we characterized the human chromosome, or the human genome, it took a very, very, very long time. So, and then it started to become a shorter period of time where you would send off a sample and maybe it would take, 6 months. And then maybe it would take 2 months. And now we can send off a sample and we get results back in 24 to 48 hours. So we have gotten much, much better at this. But I really liked an analogy that was used in my molecular genetics class, which is even if we have, let's say that we had the architectural layout of an airplane. on a map, right? So we can see how a plane is made, what its different parts are made up of. But does that tell us how to fly a plane? No. Does that tell us really what we need to know to completely understand how a plane operates? No. And the same is true for us. Even if we know what our entire genome looks like, if I can tell you every letter of your chromosome, every single nucleotide of your chromosome, that doesn't mean that I know what all of that codes for, or that it even codes for anything. So while it's useful to know what the genome of an organism looks like, and it will become more and more useful as we start to understand more, there is so much that we still can't really decipher out of that material. Okay, so I just put your learning outcomes here because there's so many of them I couldn't fit them on the slide, so you can refer back to that to make sure that we've covered all of those. The central dogma, you guys are familiar with that, so I'm just going to skip past it. As you guys know, bacteria have one single circular chromosome, very different than most eukaryotic cells. Stretched out, obviously it's really, really large. It's the same idea for eukaryotic cells. We have ways of really wrapping the DNA and making sure that it is woven really, really tightly into a very teeny tiny ball. But as we stretch it out and unwind it, it's larger than the cell itself. And that is all due to the idea of supercoiling. So you've probably heard the analogy for those of us who have used a corded phone. I don't know how many of us in the population that is anymore. But the corded phone that had the curly cord, right? It would get wrapped up on itself and tangled and all of that, and before you know it, you can't actually leave the kitchen with the phone because it's so tangled. But when it is completely elongated and totally stretched out and untangled, you could like walk to your bedroom from the kitchen and talk on the phone. That's the same idea with the chromosome. Due to that supercoiling and knotting and winding up upon itself, the chromosome is really, really tiny once it's packed into the cell. But normally it would be very, very large. Okay, just to give you an idea of the size of the genome, E. coli's genome, so all of the genetic material within E. coli is about 4.6 million base pairs. So that's a lot of nucleotides. When I say base pair, I'm talking about nucleotides that are paired to one another. Humans have 2.9 billion base pairs. But 1.5% of those base pairs actually codes for something, or we have identified something that it codes for. So humans contain a crap ton of what I like to call junk DNA that codes for absolutely nothing. So if you were to take 1.5% of 2.9 billion, how many base pairs do you think that would be? Mike, I'm looking at you. You used your math skills so well earlier.
So like 40 million?
Yeah, maybe 40 million base pairs. So really, E. coli is a teeny tiny bacterium, right, that you observe under the microscope that's like one little pink dash under the scope. And it uses all of its base pairs for everything that it needs to do. We use 1.5% of ours, so we're really not using that many more nucleotides than E. coli does to code for something. And yet we're so much more complex. So I think that's something really interesting to know.
Are there any theories on all the extra junk?
Nothing concrete. So there's definitely a chance that, some of that stuff is, like, we know that some of our genome comes from viral infections that gets incorporated into our genome over time. But there's no real concrete theory as to why we don't use most of it. It's kind of annoying, I think, that we don't use most of what we have and we just slice it out. And we'll talk about how we just cut that DNA out that doesn't code for anything as part of the transcription and translational process. Okay, the open reading frame is any region of DNA that's likely to code for protein. So, as you guys know, because of the central dogma, DNA is eventually going to code for protein, and protein's what's really doing the work of the cell. So protein is the functional product that is coming from that DNA message. So anytime we have what is called an open reading frame, that's an area of the DNA that we can look at and say that's going to code for protein. So in a bacterial cell, there is one open reading frame because We start reading a chromosome at one spot, we have an open reading frame, and the whole thing is going to code for something. Whereas in a eukaryotic cell, where not all of that DNA is actually coding for anything, we can have multiple open reading frames. Okay, just the structure of DNA, which you guys should be familiar with at least a little bit. We have DNA, which stands for a deoxyribose sugar. Deoxy meaning 2 oxygens, ribose being a type of sugar, plus a nitrogen base and a phosphate group. So you can see in each of these little sets here, we get a phosphate group, that's our PO4, very similar to ATP, right? Same phosphate group, looks the same. We have our sugar, and then some kind of nitrogen base, which is going to be our G, C, A, or T. So those are the 4 bases that are going to code for DNA. They are very specific about who they bind with. So DNA, as you guys are aware of, is double-stranded. So we see, basically, if you think of a zipper, we have the left side of the zipper and the right side of the zipper. And in the middle of that zipper are these bases that pair with one another. And they're very specific about who they like to pair with. So adenines are always going to pair with thymine. You should always see an A across from a T. That's just the way that it works. Guanines are always going to pair with cytosine. And that is just the name of the nitrogen base, depending on its structure. They're just named based on what their structure looks like. Now, one thing that you might notice is that they have a specific number of bonds, too. So, for example, adenine and thymine, we see 2 hydrogen bonds there that are keeping them together. So 2 hydrogen bonds for A's and T's, but with G and C, you see 3 sets of hydrogen bonds. So which bonds do you think are going to be easier to break? G and C, right? So we can actually characterize organisms based on their GC content. There's a manual that you guys will eventually begin to use in the lab. Let's see if I can find it. This is like the Bible of microbiology, Bergey's manual. And if I look up any organism in this manual, it's going to give me a percentage of GC content. Why do I care about GC content? I'll tell you why. Because the tighter the bonds are between nucleotides, the less likely the DNA is going to be disrupted. So if an organism has a higher GC content, that means it's going to be more difficult to disrupt the DNA. So organisms with high GC content are more likely to live in environments that are going to threaten the DNA. So hot environments, acidic, basic environments, things like that. So oftentimes we see our extremophiles having very high GC content. So that's, I think, all I wanted to say about that.
Why is a triple bond weaker than a double bond?
It's not, it's stronger.
I thought you said which one was weaker than GC.
Oh, I did say that, sorry.
Okay.
G to C are stronger. My apologies. So higher GC content can withstand more. My apologies, thank you for catching that. So GC is going to bond more tightly. Yeah.
And that's the same, so humans, we have the same amount of GC content?
Our chromosome is going to be right around the same GC content, yeah. It's not going to vary as much. Okay, another thing you might notice is that the paired DNA strands, so each of these being a strand or a side of the zipper, are oriented oppositely to one another, meaning that what you see up here on strand A at the top is a 5' end. So we have our phosphate down here at our 5' end. And then down here on what we call the 3' end is a hydroxyl group. So if you can see, right here at the end of this little purple triangle, we have an OH, or a hydroxyl. That means relatively nothing to you guys because we don't care that much about chemistry, but it's important when it comes to how these bases come together, so how the pieces of DNA pair with one another. And so when we are adding to the nucleotide structure, when we're adding DNA in the form of replicating it, we can only add to a certain end. There's only one end of this that is chemically going to be able to bond with another nucleotide. And so as we add to DNA, it's going to, how do I explain this? It's basically going to add, it's going to elongate oppositely because we have 5' end over here, 3' end over here, 3' end up here on the opposite, so it's in the opposite direction on the opposite strand, and the 5' end down here. So when we add to the DNA strand, it can only add to the 3' hydroxyl end. So when we add nucleotides to strand A, for example, if we're elongating strand A, we can only add down here to this 3' end due to that hydroxyl group. So we're going to add nucleotides here, 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 here. Whereas on this side, our hydroxyl group is up here. So we're going to add nucleotides this way, up. So as we elongate the strand, it's going to go opposite directions. Does that make sense? Hopefully? Okay. And we'll look at that in more detail. So just an overview of kind of what DNA replication looks like in an organism that has a circular structure. We have what our origin of replication in one location of that chromosome. And so the origin of replication makes sense. That's where replication is going to originate or start. So replication is going to start here, and we're going to make copies of one single DNA strand and the other DNA strand. And as we make a copy, we're basically just going to continue to unzip the DNA and make a copy all the way around until we have our two basically identical strands. So each DNA molecule generated is going to contain one parent strand and one newly synthesized strand. And so this type of DNA replication is termed, and it's in blue down here, it's the word you're missing, semi-conservative. Semi-conservative replication, because it's halfway conserved. Half of it is the parent. And we will zoom in on this picture here in just a second to make that make more sense. Okay, so if we zoom in on the replication fork where the two strands are unzipped from one another, this is what we would see. We have, as you can see, they are oriented oppositely to one another. So we have one end of one single strand that's going to be the 3' end, one end of the single strand that's a 5' end, and they are opposite of each other, right? When we make DNA, we can only add to the 3' hydroxyl end to make a new strand. So that is going to change the direction in which we can make the copy of the DNA. So it's only going to be able to go in the direction where we are adding to that 3' hydroxyl group. So just a couple of players that are important in this. In order to be able to unzip this double strand of DNA to make a copy of each side, we use an enzyme. Enzymes end in A's, right? So we know that it's going to end in A's. Can anybody tell me what the enzyme is that unzips those strands? Helicase. Does anybody know the pickup line associated? No? Oh man. I wish I was a helicase so I could unzip your genes. Use that at home tonight? Okay. So helicase is going to come through and it's going to unzip that double strand so that we're left with two single strands of the DNA. Okay? And then depending on which side we're looking at, we're going to start with the leading strand because it's easier to understand. We're going to lay down what is called an RNA primer. The RNA primer is basically added in order to, just going to move this out of the way. The RNA primer is added in order to tell the DNA polymerase, who's the one making copies of this, where to start making copies. So it's like giving it a little sticky note to say this is where copies need to begin. Then the enzyme DNA polymerase, ending in A's, is going to come in and add one nucleotide at a time that is complementary to this single strand. So it's going to come in and it's, let's say that this nucleotide right here is an A. What is DNA polymerase going to add?
T.
A T, exactly. What kind of bond is going to be there? Hydrogen. Two hydrogen bonds if it's an A and a T, right? So it's going to just lay down those nucleotides that are complementary until it reaches this replication fork, which is going to keep unzipping. So we're just going to keep unzipping that DNA, and this replication fork is going to move, and we can just continue making copies of that DNA strand. So this is very continuous and easy, follows towards the unzipping, and they kind of work with one another. As it unzips, it makes more nucleotides. So we call that the leading strand synthesis. That's the strand that's going to be moving fast and continuous because it follows in the direction that the replication fork is unzipping. Does Does that make sense why we call it the leading strand? So it's going to be faster than, say, the lagging strand. So the lagging strand has an issue. It can only add DNA on the 3' hydroxyl end as well, but because these strands are oriented oppositely to one another, then it's got to make DNA in the other direction, right? So it's working, the replication fork is kind of working against it. So it's only unzipping a little bit, and the lagging strand comes in and lays a primer and makes some DNA, and then it lays down another primer and makes some DNA, but it can only move as fast as this is moving. And it's moving in the other direction, so it's difficult for it to be able to move continuously with this replication fork. So we call that new strand, or these little strands here that are made discontinuously, Okazaki fragments. And that is just named after the guy who discovered that lagging strand synthesis is different than leading strand synthesis. So each of those little fragments is an Okazaki fragment. So we are left with all of these pieces of DNA that are discontinuous that have primers interrupting them. Because every time DNA synthesis starts, a primer has to be laid down to let the DNA polymerase know where to go. The very last step of this is to remove the primers and then fill them in with nucleotides. So once these primers are removed, we have all these little gaps where the primer was removed. So something has to come in and fill in those complementary nucleotide sequences where the primers were. So a polymerase comes in and does that, and then everything is kind of glued together so that it's one continuous strand. So we call the enzyme that does that gluing ligase. And that makes sense, right? Because a ligation is putting two things together or tying something off. And that is what is happening here. A ligase is coming in and making sure that that whole thing is continuous and tied together. So let's watch a video of that in case you're a little bit lost. And this video is found in your Connect.
Begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme
You
involved
guys
in
don't have to know the
DNA
specific
replication.
DNA polymerases.
DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Okay. Don't pay too much attention to that last 10 seconds. I don't want to get that complex. But basically what I wanted to show you is how the lagging strand differs from the leading strand. So the lagging strand is adding nucleotides away from the replication fork, and it can only add in those discontinuous segments called Okazaki fragments, whereas the leading strand is moving towards the replication fork, so as it unzips, it can just follow that unzipping. Does that make sense? Let me know if you have any questions about that. Yeah.
Could you just, you can tell me no too, because I had a hard time understanding this stuff. Like, I always thought it would be easier to understand if we just wrote out like a list,
Okay, sure.
a step-by-step list.
Sure. So I won't tell you no, but I will tell you that you're right here. You have this list 1, 2, 3, 4, 5, 6. If you write that out in order, that might help you. It would take me a really long time to get that on the board.
Yeah, no problem.
Cool. Okay. All right. So now we move from the central dogma from DNA replication to transcription, which is taking that DNA message and transcribing it into mRNA. So mRNA, or messenger RNA, is what's going to carry the code to eventually make a protein. It's very similar to DNA, actually. The only difference is that instead of deoxyribonucleotides, we just have ribonucleotides. So the sugar is not deoxyribose, but ribose. The other difference is that there is no thymine found within RNA. Instead of thymine, uracil is present. So anywhere you see a T, you replace it with a U. It's pretty much that simple. To make new RNA strands from the DNA message, we use an enzyme called RNA polymerase. So just like to make new DNA, the copy machine is called DNA polymerase. To make a new copy of RNA, it's RNA polymerase. It's pretty simple. It's an enzyme, ends in ase, so you can remember it that way, that it is an enzyme. But the difference here is that no primer is required. So RNA polymerase knows where to start synthesis of RNA. It doesn't need a primer laid down in order to tell it where to begin. So that makes things a little bit more similar. You can think of RNA as being an intermediate between DNA and protein. So it's going to be the one that's really going to send the message of what protein needs to be synthesized. So it's kind of the middleman. So there are 3 types of RNA. The one that is actually going to eventually translate into protein is going to be the messenger RNA. The messenger RNA is complementary to the template sequence of DNA. And we'll look at DNA here in just a second, look at how RNA compares to that. But it is complementary to it. So it has the opposite message, basically. And those nucleotides are going to be grouped together in groups of 3 called codons. So every 3 RNA nucleotides equals 1 codon. We can then translate that codon into a protein message. So that is mRNA. tRNA, on the other hand, carries what we call the anticodon. So tRNA is what is going to actually act as the wingman, basically, between the RNA and the amino acid. So if in the central dogma we have RNA that's eventually going to be translated into amino acid, it's the tRNA that brings them together and decodes the RNA message into the protein. So you can see the structure here. The way that we have it kind of drawn out in cartoon form here, it kind of looks like a T. Structurally, not so much. This is what the actual structure kind of looks like. But when we draw it out, it kind of looks like a T. And we call it tRNA because it is transfer RNA. So it's going to act to transfer amino acids to one another in order to make a protein. So down here at the bottom of the T, at the bottom of the structure, is where the anticodon is carried. So, for example, the anticodon that we see here is UAC. Where are my markers? UAC. So we have UAC. The anticodon is going to be complementary. complementary to a codon on the messenger RNA. So if we have our messenger RNA sequence, that's gonna eventually code for a protein, and it's got all of its codons, it's going to have a complementary sequence to this anticodon, UAC. So what is complementary to UAC? Let's see, what's
A.
complementary?
A.
Good.
T,
But not
U.
T because we're
U.
mRNA, right? U, good.
G.
G, excellent. Blah, blah, blah. So this is the anticodon to the codon in the mRNA. So the whole reason that we use transfer RNA is because this transfer RNA on the other end of it, you'll see, is what is called the amino acid attachment site. So if I, draw my T blob, whatever that looks like, up here I have my amino acid attachment site. So literally, the amino acid that is coded for by AUG is going to be attached to the tRNA up at the top there. Does anybody know, just off the top of their head, what AUG codes for? Methionine. So methionine is just the name of the amino acid. So it's going to be carrying that amino acid on the top of the tRNA. So it's really acting as the wingman between the amino acid and the RNA codon. And it finds the RNA codon that it needs that corresponds to that amino acid based on its anticodon. Okay? So that's what tRNA does. And then ribosomal RNA is the final type of RNA that is a component of the ribosome, which, go figure, is where protein synthesis occurs for translation. So these are all involved in transcription and translation. And we will look more at the structure of the ribosome here in just a few when we talk about translation. Okay, so there are 3 steps that kind of happen in transcription: initiation, elongation, and termination. So initiation, how does transcription get started? Elongation, this is the actual process where the nucleotides are being added to make the RNA sequence that is complementary to the DNA. And then at some point it's got to stop because it has a code within the DNA that tells it, or within the DNA that says, okay, we're done transcribing this particular set of genes, so we can finish. So there are a couple of different ways in which transcription can terminate, which we will also talk about. So initiation is going to begin where RNA polymerase binds to what we call a promoter sequence. So, geez, I left everything over here. So transcription knows, or the RNA polymerase knows where to begin transcribing a piece of DNA thanks to a promoter sequence telling it where to go. So whereas DNA polymerase requires an RNA primer to know where to start, RNA polymerase requires a promoter sequence to know where to start. So this is kind of a busy figure, but it's showing the entire process of transcription and what's happening. So up here is our DNA sequence, double-stranded. So we have our 5' end and our 3' end. You'll notice they're oriented oppositely to one another. So this is our double-stranded DNA. And within that DNA sequence is an area called the promoter sequence. So this is a set of nucleotides that is called a promoter because it looks a certain way. So it has a certain order within its nucleotides that we look at it and we say, okay, that is a promoter. That is calling RNA polymerase to the area. Then RNA polymerase recognizes that promoter sequence and it begins transcription. And we'll come back to this figure over and over again as we go through and go through transcription.
Can I ask you a question?
Yeah, sure.
I think you just said that the promoter sequence is in the DNA, just transcription, not the M or T RNA?
Correct. The promoter sequence is buried within the DNA. Because the whole process, what we're doing is taking our double-stranded DNA and we're making that into an mRNA. So RNA polymerase is looking for a sequence in here to say, okay, where do I start making my RNA strand from this? Does that make sense? So it could start at any point within this DNA and start making RNA from it. but it needs to find the area where transcription really begins. So let's say that, for instance, we want to begin transcription of gene, we'll call it gene 1, gene 2, and gene 3. Maybe these are genes for a flagella, okay? So if it needs to transcribe the genes for a flagella, it's got to figure out where those genes are located. So it's going to find those genes based on the promoter sequence and know, okay, I need to make the mRNA message of these 3 genes by finding the promoter sequence. Okay? So if we zoom in on that figure and we look at the DNA, this area called the promoter sequence is going to be found right before where the genes are coded for. So the genes, remember, are embedded within the DNA, but they don't mean anything until they're made into RNA to be made into protein. So they are here, but they have not been transcribed yet. So we have to know where to start transcribing, and that is how we use the promoter sequence. The promoter sequence here that you see is what we call a consensus sequence. So a consensus sequence means that these nucleotides have a very similar order in which they go in. So what you see here is TD-GACA and TATA. That's how you can remember it. TD-GACA and TATA are 2 nucleotide sequences that are a certain distance apart from each other, and they're located in a certain point in the gene, and they say transcription starts here. So the promoter sequence is a flag saying start here, and the RNA polymerase recognizes that. Okay. So the RNA polymerase finds this promoter sequence thanks to what is called its sigma factor. So I know this is a lot of new terminology, so we're going to work through this kind of slowly. The sigma factor recognizes the promoter sequence, and that's how RNA polymerase decides where to go. All right. So what's also important to note about this is that this could be what we call an operon. An operon meaning that several genes are under control of the same promoter. So we want to transcribe more than one gene, but we only have one promoter that tells that RNA polymerase where it goes so that we can transcribe all those genes at once. That would be called an operon. So we will learn about the lac operon and the ARG operon probably tomorrow, and that's going to be a bunch of genes that are under control of the same promoter sequence. This promoter sequence, this consensus sequence that you see here, is going to be very similar across the board within DNA. So it could be off by 1 or 2 nucleotides, but for the most part, this is what a promoter sequence always looks like. Okay? Okay, so let's break that down. We have the sigma factor, which is just a piece of RNA polymerase. So if I drew the enzyme, RNA polymerase, it's just a big blob. Okay, it's a protein. That's what enzymes are made up of, is mostly protein. And it has a piece on it at the bottom called the sigma factor. So sigma symbol. This is the sigma factor. The sigma factor has eyes for the promoter sequence. It is made to recognize that TD-GACA TATA box. That's what it does. It looks for that sequence within the DNA. Once it finds it, it tells RNA polymerase to go to that promoter. It's kind of like a GPS that tells the RNA polymerase this is where your promoter is located. So that's the first thing that happens. Then RNA polymerase binds to that promoter sequence. And very similar to DNA replication, the DNA needs to unwind into its single strands. As a double strand, with those nucleotides hydrogen bonded to each other, you can't do anything with that. You have to unzip it so that you can make copies of it. You can't make copies of both strands at the same time. So the first thing that happens after the RNA polymerase binds to that promoter sequence is unzipping. So the DNA separates into each of its single strands, And from there, the RNA is made complementary to the template strand. So around 9 nucleotides are made at first. Again, does not require that a primer is laid down. And then the sigma factor leaves. So if we come back over to this figure here, sigma factor and RNA polymerase recognize the promoter sequence, they lay down here, and transcription begins. The DNA is unwinded into each of its single strands. We call one of those strands the coding strand, and one of those strands the template strand, which is back on a figure a couple slides previous to that we'll revisit. So it breaks apart those strands, we get 2 different strands, and now the RNA polymerase can start making RNA that is complementary to the template strand. So it's going to lay down nucleotides that are complementary to that template strand. So if the template strand has an A, what is RNA going to lay down?
U.
A U, right? Not a T because it's RNA, but a U. So if it's an A, it's going to lay down a U. If it's a T, it's going to lay down
An
A, and so on and so forth. And it keeps making a strand that's complementary to that template strand. And that is how we get our mRNA. Eventually, after 9 nucleotides are laid down, then the sigma factor actually dissociates and leaves. And then it will go find another RNA polymerase to work with to help guide it to the next promoter sequence. So they're not necessarily attached to each other forever. It can help other polymerases to find its promoter sequence. So that is the process of initiation. So elongation is just that RNA polymerase going through and making copies of that template strand, just 1 nucleotide at a time until we hit a point in which things need to stop. So as this happens, very similar to DNA replication, the DNA strand is reannealed behind it. So it goes from being 2 single strands to being zipped back up behind it, just to make sure everything remains kind of tight and clean. So it's going to zip the DNA up as it's not needed anymore. What's interesting about transcription is that it can happen simultaneously. So as soon as the big promoter enzyme is out of the way, another promoter, or the big RNA polymerase enzyme is out of the way, another RNA polymerase can come and lay down and start again. So we can transcribe the same gene several copies at a time simultaneously. Okay, how does it know how to stop once it starts making that RNA sequence? There are 2 ways in which RNA polymerase finds out that it's done transcribing those genes and that it can be finished. One is called a hairpin loop. So basically, the DNA, or I'm sorry, the RNA zips up onto itself. So it's complementary to itself, and so it can come and it can just bind to itself, and it forms what we call a hairpin loop because it looks like a hairpin. So as you can see, this was originally one kind of, let's see. So it's basically one strand. This is our RNA strand. But these nucleotides here end up being complementary to these nucleotides, and so it just zips up on itself and forms a hairpin loop. RNA polymerase obviously can't do anything with a hairpin loop, so it stops and it falls off, and it can go on to transcribe some other gene. But essentially transcription is terminated at that point. So whenever that hairpin loop forms, everything stops. That's one way this can happen. Another way that it can terminate is through what is called the Rho enzyme. And so this is just an enzyme that wraps around the RNA. And this makes the RNA become unstable, and the RNA polymerase can't work with unstable RNA. So again, falls off, and transcription is terminated. So 2 ways that it can end. How does it know where to start? Promoter sequence, good. So the promoter sequence tells RNA transcription where to start, and termination occurs either through a hairpin loop or through an enzyme. Okay, so now that is transcription. That's the process in the central dogma of making DNA into RNA. Now we want to take that RNA and we want to make a protein. So how do we read that RNA and make protein out of it? Through those codon sequences. So through that triplicate set of nucleotides that's going to code for each single amino acid. The sequence of those codons is what is going to determine the sequence of the amino acids and how they come into order with each other. The amino acid sequence is the basic structure of the protein. And so you change that sequence, you change the protein. You change the sequence, you completely change the structure of the protein. The structure of the protein is what determines its function. So as we learned with the prion protein, whose function we have no idea what it is at this point, but we know that if it's folded wrong, it completely malfunctions and starts to cause disease, right? So the structure of protein is so important to its function, and as soon as you change that structure, you change its function. So it's very important to note that that amino acid sequence is very important for the actual function of the protein itself. So this is just a little bit of biomath. If we know that there are 4 different kinds of nucleotides that can be in RNA, so we have our adenine, our uracil, right, and then our guanine and cytosine. 4 nucleotides, each codon is 3 nucleotides long, then that tells us we have 64 potential codons. 64 different ways that those nucleotides can be in a certain order, just statistics, okay? So 64 possible codons, and yet there are only 20 different amino acids that make up proteins. So what does that tell you? There are less amino acids than there are codon sequences. So that means there's more than one codon for one particular amino acid. So when you look at, we'll use a codon table. When you look at that codon table, there are several different potential combinations of nucleotides that might code for one particular amino acid. 3 of these 64 codons are what we call nonsense codons, meaning they code for nonsense. They don't mean anything. So 3 combinations of these nucleotides do not code for a single thing. That's how translation knows how to stop. Because the tRNA finds something that it can't work with. Okay, so this is the genetic code that I was talking about. So if we look at these, each of these 3 nucleotides in order, in a particular order, these are what we call a codon. Each of these is a single codon. So there's 64 of these guys. 3, 1, 2, let's see, 1, 2, 3 of these code for absolutely nothing. So we call those our nonsense codons. And a lot of these have more than one codon that codes for one particular amino acid. Any questions about this?
You don't want me to memorize this table?
None of these do you need to memorize. Nope, you'll be provided with this table on your exam.
I had to memorize.
It will help you to be able to recognize a couple of the important ones. For example, AUG is the start codon. So we'll talk in a second about how translation knows where to start. It knows where to start because it's going to run into an AUG. So it's got to find that first AUG start codon, and then translation continues on from there. And then it continues and continues and continues until it hits a stop codon, or a nonsense codon, something that it cannot, tRNA cannot work with. So UAA, UAG, or UGA. So you don't have to memorize those, but be able to recognize them within a sequence at least so that you can kind of get a feel for where the start and the stop codons are located. Okay. So the key players in translation, obviously the RNA is going to be a key player because it's what's going to be translated into the amino acid sequence, so we need to use it as our template. Ribosomes, our little protein factories, are going to be really important in protein synthesis. And then our tRNA, our wingman, who's going to bring together the RNA code and the amino acid. Okay, so I'm going to skip past this for a second. And 1 second. I want to actually just go and practice this real quick. So I'm going to pass these out, and we're going to practice this so that we can start to put it into context. And then I'll let you guys go.
I like this stuff. I like it too.
You should do some kind
Hard to memorize that table for my
That's
bio
just
class.
so silly.
Isn't that crazy? Certain phonetic sequence.
Okay, so really quickly, I want to revisit
There's no use.
I want to visit this slide. So what you are given here is the coding sequence. So you're only given one strand of DNA. It's really important that you know which strand of DNA you're working with, because as I've shown you in this figure, after transcription begins, the DNA is going to split into two strands, the coding strand and the template strand, right? The RNA is going to be made from complementary to the template strand. So depending on which sequence I give you, it's going to be really important to determine what your RNA sequence looks like. So coming back to this figure, we have our double-stranded DNA here. The top strand that is given to you 5' to 3' is the coding strand. So right away you can write on your worksheet, give yourself the orientation. So you know 5' is going to be on the left side and 3' is going to be on the right side. So I'm just going to write out this sequence really quick. So right away, if I tell you I'm giving you the coding strand, then you know it's 5' to 3' for the orientation. The bottom strand is not given to you. That's the, yes.
Sorry, I just have a
No, you're
question.
fine.
On the third one, are you wanting the sequence of the mRNA molecule of the one I just wrote, or the one that you gave us? On the third one, or on the second one, sorry.
Write the sequence of the RNA molecule that corresponds to the DNA. So it's gonna, so that's what I'm going through is, it's gonna be, if this is our coding strand,
Yeah.
the RNA is not made from this, it's made from the template strand. So it's gonna be
Oh, okay.
complementary to the template strand, which you haven't been given. But
Okay.
you also know that the DNA, double-stranded DNA is complementary to each other, right? So the bottom strand, even though it's not given to you, you could come up with it. So the template strand is just going to be complementary to the coding strand that I gave you. So if you want to write that out, you can. And this is just based on how nucleotides pair with one another.
So why is that C bolded and underlined?
We'll get there.
I'm sorry.
Don't worry, you're good. Okay. So now we have our template and we have our coding. Now if I wanted to come up with my RNA strand, which is, let's see. So number one, we wrote the sequence of the template strand. Number two, write the sequence of the RNA that corresponds to this. So now all I'm going to do, if I know RNA is complementary to the template strand, I just write the nucleotides that are complementary to this template strand. But keep in mind, there are no thymines. So instead of thymine, uracil. So this A is going to be a U. A, U, G, C, and so forth.
So you essentially go back up to the coding one. and just
Exactly.
substitute the
Yep,
C.
that's where we're going. Yep.
You have to start. Where's that? And then
Okay, so does anybody have any questions about how I came up with my RNA strand and what that looks like? So I just went through every nucleotide of my template strand that I made, and I wrote the complementary nucleotide to each of those. So here, normally it would be a T, but because it's RNA, I've got a U instead, and so forth.
The last U and the three
Oh, yes, thank you, sorry. Nice catch. Good catch. Okay, so what do you notice about this RNA strand in comparison to the coding strand? It looks exactly the same, except instead of T's, I have U's. Because the template strand is complementary to the coding strand, and the RNA strand is complementary to the template strand. So shouldn't these be the same? So that's why I told you for number one, it's optional for you to write the sequence of the template strand. Because you don't need it to come up with the RNA strand unless you like to do it this way. Otherwise, you could just copy this baby down and replace your T's with U's. Okay, so that is transcription. We just carried out transcription. Imagine there is a Tauta box back here, or a TDGACA sequence. So we carried out transcription and now translation. So we only have 1 minute left, so I'm just going to point out a couple of important things and then I'll let you go and we'll finish this up when we come back. How am I going to know where this begins? Do I just start breaking this up into threes? No. How do I know where to start?
the AUG.
I gotta find my AUG. So for simplicity's sake and being able to do this on a worksheet, you just find the first AUG you come across, okay? AUG, right here. And then from there, what I like to do I just like to underline all of my triplets so I don't get mixed up. Okay. So the next step from here, I'm going to go head now to my codon table. I know AUG codes for a methionine. But in the case of the first AUG of the entire sequence, which codes for a stop codon, in bacteria, that very first start codon is modified a little bit. So it's what we call a formal methionine. Not formal like a formal dance, formal like a chemical. FORMYL. So formal group. That's just a chemical group that's added to the methionine to say this is where translation starts. That's just the autoclave stuff. Okay, so we've got our start codon. Then from there I can start matching these up. CUA. Okay, so I look over here. First base position, C. Second base position, U. Third base position, A. So, Lucy. GAC. G. Second base position, A, C. Aspartic acid. Any questions on how I did that? Okay, so last thing I'm going to say before I let you guys go is that at some point I'm probably going to find a stop codon. So I just know off the top of my head because I have my codons familiarized. UAG is a stop codon. Here's my UAG. So this is where everything's going to stop. So I would come up with, the rest of the nucleotides and then stop. Any questions I can answer? If you have time, I'll stick around for a few minutes if you have any questions that you want to come up and ask, but other than that, you guys are free to go and we'll pick up on this tomorrow.
Probably.